Good morning, friends. Yes, myself, your botany mentor, Dr. Mukesh Kumar. Most welcome in your dream classroom, Hortus Botanica for all the medical aspirants. As you all know, we have started plant physiology series. With that, we are continuing the transport in plants, and yes, already we have conducted lecture 1, lecture 2 of plant physiology, and today we will discuss the lecture 3, that is osmosis. A very common term, osmosis, associated to plant water potential, DPD, diffusion pressure deficit, osmosis, also related to plasmolysis, turgidity, means almost all where we are studying the water plant relationship. So if you are going to study the about your osmosis, so you all know this osmosis is a special type of diffusion through semi-permeable membrane. And we have the plant cell have cell membrane covered by cell wall, and this cell membrane is also executing the special diffusion that's called, yes, osmosis. We must know about the role of water when it will go inside the cell, how they are moving, and how it's very essential for the different type of the plant physiology, that is maintaining the vitality of a plant cell, isn't it? So have a look about the, yes, about the this one, that is your plant cell. So we are talking about plant cells. Say this is the leaf margin, and inside what we have, yes, we have this hexagonal plant cell. See the brain of the cell, nucleus, and nucleus controlling part of the cell. Inside nucleus, yes, we have, see the nucleolar membrane, we have the nucleolus inside, and this is the site where ribosomes are forming. Ribosome is a protein factory, and see how they are coming outside in cytoplasm and synthesizing protein. That is very important for the enzyme, hormone secretions, metabolite formation also. See the main arena of the cell, that is the cytoplasm. In the cytoplasm, we have the endomembranous system, and first member of that is the endoplasmic reticulum. See, we have the smooth and the rough endoplasmic reticulum. In the rough, we all know no ribosomes we have, and when ribosomes have, then rough. This is the rough. Okay, and this is the traffic police of the cell. Yes, my dear, this is the Golgi complex, and they will form from the endoplasmic reticulum. After that, we have the vacuoles also, lysosomes also. These are the member of endomembranous system. And see the powerhouse of the cell, mitochondria, that perform the cellular respiration. And cell wall, the main rigid out guard of the plant cell. See, this is the lipid membrane structure, and see they have the plasmodesmata, that is connecting two adjacent cells and exchange the cytoplasmic material. So see, total aqueous system, we have water inside and outside, and how they are moving inside when it is being required. So this is very essential. Now see the whole membranous structure inside the cell, and water we need. Without that, we can't perform any cell physiology. Yes, this is your chloroplast. This is the kitchen of the cell that will perform photosynthesis. They have the grana. See, these are the tracks of the grana, will trap the sunlight and perform light reaction after that dark reaction. And we have the glucose, and see the vacuole here, vacuole membrane covered by the your tonoplast. Okay, that is also the semi-permeable membrane, and the cell membrane and this tonoplast, this is the determinant for the osmosis. Okay, and see how we have the plant cell. So we must know these plant cells because they have the membranous system, cell membrane they have, and they are participating in the osmosis also, because we need water, isn't it? Yeah, so by this all we have understand about the importance of this osmosis. Now mates, the very great scientist, yeah, knowledge, and the knowledge Yeah, he first time he discovered the process of osmosis, and he discovered in the pig's bladder, right? Now, see what is osmosis. So before telling the definition of osmosis, I would like to give you the concept of osmosis so that you can define osmosis in several ways. For that, see, get a demonstration first. What happened? I have taken here a U-shaped tube. So this figure, and this is a U-shaped tube. Here see how this two side A we have, and here B we have, and both is being separated by the semi-permeable membrane. Now, in A side, discuss what happened. We have a solvent. Suppose water we have taken, and we have this solute, and suppose anything you can take either sugar or salt, and they are performing the solution. See here also side B, we have this solution made up of this solvent as well as the solute. But big comparison, what we are finding here, we find here that B side we have more solute concentration. See, this is the highly concentrated solution. Amount of the solute is more in B. So here B is high, solute concentration is high. Since this is high solute concentration, so the solution is known as hypertonic. Similarly, in A side, we have the low concentration of the solute. So this is solution, and that solution is called as the hypo, means low, hypotonic solution. Means to say, we can say that a solution is being known by its solute concentration. That's why it is the hypotonic and it's a hypertonic solution. So we have created a very good osmotic active solution. This is called OS, isn't it? Now what happened in this condition? What will happen? See, here we have got solvent, and this solvent is low with respect to here. Yes, here solvent is high, but remember, solute is low, right? So here also, as I told you, this is your hyper, means high. So this high concentrated solution, but this is your low concentrated solution. So what will happen? Yes, see this arrow is indicating that the water solvent will move from A to B. So what you are finding, this movement of solvent or the water through this semi-permeable membrane, my dear, this is a special type of diffusion, and yes, this is called osmosis. Now let's discuss about the definitions. Okay, see, if you are going to define in terms of solvent, then what will you tell? Yes, this is the movement of water from high solvent to low solvent through semi-permeable membrane. Second definition, you can say, what is osmosis? Yes, this is the movement of water from low concentrated solution, that is hypotonic solution, to high concentrated solution, that is the hypertonic solutions, through semi-permeable membrane. Similarly, at A side, we find here diffusion pressure deficit is low, and if this is low, the water potential is high. We'll discuss in detail in further lectures what is dPd and water potential. But here what we have, since the solute is high, so dPd is high, OP is high. Just opposite, yeah, water potential low as well as solute potential, this is also low. So we find here that in terms of the dPd, you can say this is the movement of water from low dPd to high dPd, or high water potential to low water potential, isn't it? Also in terms of the osmotic pressure or solute potential, we can define in the same way. With the osmotic pressure, it's low to high, or solute potential, it's high to low. Isn't it? So this is the just basic concept about the osmosis, and you can define in several ways. So let's discuss what NCERT has given. See, this is the movement of solvent, and yes, this is from the hypotonic solution to hypertonic solution, and this is through semi-permeable membrane, just I told you. So what this is, yes my dear, this is a special type of diffusion of water, pure solvent, no, and through the semi-permeable membrane, SPM. This is your semi-permeable membrane. So This is also supposed to be what? A movement. This is a movement of water from low chemical potential, that is concentration, okay, to high chemical potential. Yes, that is through semi-permeable membrane. So you have seen, if high chemical potential is there, means to say they are supposed to be the hypertonic solution. This is your definition in NCERT. And low means to say, yes, this is your hypotonic solution. Right? So from the hypotonic to the hypertonic solution, how they are moving, this is the osmosis, right? And see, I describe you about their demonstration. Now, we have two important factors. First, membrane, we discuss about the semi-permeable membrane. Second, we are talking about where they are moving. And for movement, we have two type of the solution. One, we have taken the cell. Inside the cell, what we have, as I told you, this is your cell. So inside the cell, what we have, the liquid inside the vacuole, this is supposed to be the cell sap. And outside, what we are putting, this is supposed to be what? This is supposed to be the external solution. And on the basis of this external solution, okay, it is decide what will happen. Either plant will go inside or plant will allow the water outside. What will happen? So this will depend upon the concentration of the external solution. Concentration means tonicity. Tone means volume. Concentration. So tonicity of solution, and that is solution means to say, yeah, external solution. So on that basis, we found that we can take three type of the external solution. First, it may be the isotonic solution, it may be the hypotonic solution, or it may be the hypertonic solution. As the term suggesting, iso means to say same, and tonic means to say concentration. So what we find here? Yes, what is the concentration in cell sap? That is the also concentration of the external solution. Yes, my dear, means to say the solute concentration of cell sap inside the cell is equal to the external solution. So what happened? This cell is called as the flash-sheet cell, and the movement will be maybe going inside, again coming outside, again going inside, coming outside. So see how they are showing this reversible process. They are moving and coming. This indicate this is the isotonic solution, and the nature of the cell is called, yes, plastic shell. Now talk about the second part, my dear, and this is the hypotonic solution. Hypo means low and tonic means concentration, just we discussed. So outside we have what? Yes, see this outside we have the low concentration. So if outside we have the low concentration, then what happened? Inside we have the high concentration. So what happened here? See the concentration of cell sap inside the cell, it is greater than what? External solution. It is greater than external solution. So what will happen? Inside we have high D. So it will absorb water from the outside and the movement will go in. And this process is called endosmosis, leading turbidity. And the cell, what happened? It will swells up, and this swollen cell is called turgid cell. Okay, so question may come to you that turbidity occurs in which condition? Yes, one and only condition, that is the hypotonic solution. Similarly, when we talk about the second case, that is the third case, that is the hypertonic solution. Hypo means to say high, tonic concentration. So here what we have? We have the high concentration outside. So what we have? Cell sap concentration less than the external solution. So what will happen? Just opposite. Yeah, the water will come outside, exit, and as a result this process is called exosmosis, and the cell size will shrink down. And this shrinkage of cell is called plasmolysis. Okay, and similarly the swelling of cell, and the swelling of cell is called turbidity. So in this way, my child, we find how we had the three types of the external solution. Yes, isotonic solution, hypotonic solution, and the hypertonic solution. Equal, then flaccid condition, outside we have low. So what happened in osmosis? It will lead turgidity, and cell is called what? Turgid cell. Here we have the plasmolized cell, right? Now, second, as I told you, the cell membrane, they have very important role. We must know about the cell membrane, how we can feel it, absorb it. Yeah. See, if you are talking about egg, it is also given in our NCERT, right? In egg, outside what we have? We have cell. Then inside we have the cell membrane. So if you want to remove this cell, then what happened? After removal, you have two things. One, yolk, and second, white part, that is called albumins. So in order to remove, what happened? We are using HCl commonly, and then what happened? It will dissolve. So what will you have now? You have only the cell membrane of the egg, and this is supposed to be work as the semi-permeable membrane. Means to say, by this also, if you are allowing the water, making them the hypo and hypertonic solution, so they will also become like the membrane, and they will reflect the same things what we studied. In hypo condition, what happened? They will swell up. In hyper condition, they will shrink down. So we can see it very clearly right now. So yes, I want to show you also through a very good demonstration here. So see the demonstration. That demonstration will tell you about what? Yes, that demonstration will tell you about this all osmosis, endosmosis, exosmosis, swelling of cell, as well as the turgidity of cell. Okay, now have a look. See, little faster. See, this is the dissolve, and what white membranous part, this is the cell membrane, and that is yes my dear, that is your semi-permeable membrane. We have dissolved it. Before that we can weight it and see hypertonic, and the hypertonic solution too we have taken. And see, in first case what happened? Yeah, in the hypertonic it will shrink down, in hypotonic it will swell down. Two cases we have. First hypo, swell down, and then again weight, how the weight will increase, and one weight will down. So it is reflecting at how they are executing what? Yes, isotonic solution and the hypertonic solution, isn't it? Yeah. Now let's see my child. This is the NCERT diagram, same tonicity of solution. First case, water is moving outside, and this is the demonstration of plasmolysis. The cell is called plasmolyzed cell. See here what is going inside and outside, and this is, you can say, yes, this is your flaccid condition, flaccid cell it is, no? And here this is a turgid cell when water is going inside and it will swells up, right? Now, again, I want to show you the very good demonstration, my dear. See, demonstration of the osmosis again, right? In this osmosis, I will tell you again, see, what we have, the semi-permeable membrane. And see, one right side, we have the hypertonic solution. So, see how water is going from hypo to the hypertonic solution. Yes. See, the brown membranous is the supposed to be the semi-permeable membrane, and see towards the hypotonic water is moving, and as it is moving, it is creating a pressure, and this is called osmotic pressure, isn't it? Yeah. So I think it's also very much clear. So by this all supplement diagram, I think your concept is very much clear. What is the hypertonic solution? What is hypotonic solution? How we can also create it in our daily life also, right? Yeah. So okay, now let's see. So just I discussed you what I discussed. Yes, what is definition of the osmosis? Okay, then I show you different terminology. This is the external solution, three type of the solution. Then how we can make egg as a source of the semi-permeable membrane, and we can execute their hypertonic and the hypertonic solution. Then we discuss about three type of the cell: plasmolyzed cell, flaccid cell, turgid cell, and this demonstration also. Now let's see what happened in osmosis. Just I told you, this is the movement of water and the solvent through semi-permeable membrane. So it will move from, yes, from low DPD to high DPD, from low OP, OP means, yes, this is your osmotic pressure, and this is your water potential, right? and this is your solute potential. In terms of solution, it is movement of hypotonic to hypertonic. So see, this is your one definition, this is your two definition. Here it is two definition in terms of water potential or solid potential, and here again you have the one definition. So you can define osmosis in terms of how many ways? 6 ways minimum, isn't it? Yeah. Now, what are the characteristics for osmosis? Yes, if we are talking about the plant cell, what we have? See, we have the plant cell and is being covered by, yes, outer membrane. This is your cell membrane, which is enclosed by the cell wall. Inside we have the cell membrane, and see, inside that, what happened? This is your tonoplast. And tonoplast is the cell membrane of this vacuole. Yes, this vacuole is the cell sap. So you find here this cell sap and the tonoplast, it will constitute the vacuole of a cell. So what happened, as you know that this cell wall, this is your cell wall and it is a freely permeable, easily anything can go inside. But cell membrane as well as the tonoplast, what are they? They are the semi-permeable membrane. Mind well, semi-permeable membrane. So if they are the semi-permeable membrane, so what will happen? They will decide that water will go or not inside. They will determine. So they are the determinant of for the water movement, isn't it? Question will come, or question has come in previous NEET examination. Now, so this osmosis depends upon the OP, or also called as concentration gradient, as well as, yes, target pressure or pressure gradient, TP. Okay, so water always moves from higher water potential to lower water potential. See, this is the two problem and this is the two problems at the rate of your NCERT, at the rate of NCERT. What the two questions they have given? Question number 1. Okay, in first question they are asking that, see, this is your cell A and this is your cell B. In first case, this is your water potential, always found in negative value. This is the megapascal, okay, pressure. So you are finding that here the water potential is 2000, and here what you are finding, this water potential is 1000. This is in minus. Mind well. So which is more? -2 is more or -1 is more? Tell me. Yes, my dear, you are correct. -1 is more. So -1 is what? Yes, -1 is the higher. Okay, so this is your high water potential and this is your low water potential. And you all know water move from, yes, from the high water potential to low water potential. So question will come, where the water will move? Yes, water will move from B to A. Similarly, see the question number 2 given in our NCERT. Gita, what? First, suppose this is your X and this is your Y. Two cell we have. In first case, we have -0.1. Here it is -0.2. Just I told you, minus is there. So this value is low, this value is high, and water potential always move from, yes, my dear, high water potential to the low water potential. Isn't it? Yes. Very simple. Now, at equilibrium state, very important point. What happened? Water potential of A and B are nearly same. If they are equilibriums, of course it will be same, right? Now see how we can create the pressure so that water is going inside. In fact, when you are taking, see, this is your a tube I have taken here, and inside we have created a solution. Inside the solution, here we have taken sucrose and the water is there, so sucrose solution outside, but we have the pure water. So what happened? This inside is supposed to be the hypertonic solution and outside we have the, yes, my dear, it is a hypotonic solution, and they are being separated by the semi-permeable membrane. So what happened? Of course, they will tend to move inside, mind well. But if they are not going, mind well, if they are not going, means to say, see, their arrow is given that they are not going, they want to go. So that pressure that is making them to stop, this is also supposed to be the osmotic pressure, and this is the osmosis also. When this pressure is little now higher, then they will go inside, and the pressure, that extra pressure is supposed to be what? OP, osmotic pressure, isn't it? Now see further. Again, I want to show you how this is very important, my dear, this osmosis, and they are performing very important cellular activities. So let's see. Here we have again cell we have, and we find that this water plant we have, and see how water is moving. See water on the leaf surface. See, it is the cell, leaf cell, water we need for photosynthesis. So what do you find? See how water are being taken inside the cell, because cell has the cellulose, and that is your chloroplast. Chloroplast will perform the photosynthesis, and due to photosynthesis, what happens? They have the glucose, and glucose will make them hypertonic solution, and that's why it will move from hyper to hyper. See another example demonstration. See a very simple beaker is taken, two side equal concentration is there. This is the equal concentration, equilibrium state. Now right side we are adding urea. By addition of this urea, what happened? Right side it become hypertonic solution, but left side, yes, this is the hypotonic solution. So what happened? Yes, a pressure gradient will create, because right side we have the hypertonic and left side we have the hypotonic. So hyper will create a gradient. As a result, hypo will move towards the hyper. Let's see, a gradient is going to be create, and finally, yes, it has been created. And see how hypertonic water molecules are moving. Yeah, they have started to move. Yes, and how they are moving? See, the level of the water will decrease gradually, as I told you, and this is called what? Yes, this is called as the OP, osmotic pressure. See, left side decreasing, and proportionately right side it is increasing. So again, this is a very good demonstration of osmotic pressure also, as well as osmosis. So see, similarly we can feel, yeah, this is the hypertonic solution. That's why it happens. This is moving from the hypertonic to the hypertonic solution. Okay, and it is moving here, right? So again we found here, this is a movement, and see again we have taken a cell. Outside this water molecule will come H2O. So see, yes, so this is finding, showing you turbidity and the plasmolysis, what I discussed you. What we are finding? See, water molecule outside high concentration is of water, inside we have low concentration, right? Right, so water concentration is high, so it is moved. Solution is hypertonic inside, outside hypotonic. So see how water is moving inside towards hypotonic to the hypertonic solution, or solvent level you can say hyper to hypo, right? So yes, my dear, we find here how different mode we have to demonstrate and make you understand that this is moving from the hypertonic to the hypertonic solution. Now on the basis of all these analysis, we find, yes, my dear, we have two types of osmosis. One is the exoosmosis. Yes, what is this? This is the osmotic withdrawal of water from cell. See, this is the osmotic withdrawal of water from cell. And when it will happen? Yes, repeat again. Yes, who will tell me? Nice, my dear, you are correct. This is because of the hypertonic solution. And when there is the osmotic entry of water into a cell, this is called the endosmosis. And this is, yes, who will tell me? Yes, you are right you are. This is due to the hypotonic solution, isn't it? Very good. Now let's see about the osmotic pressure. This term was given by paper, and this is the pressure required to prevent water from the diffusion through semi-permeable membrane. I told you here they are going to prevent, and that preventing force is supposed to be the what? OP, osmotic pressure, my dear. And this will depends upon, yeah, first and very important, solute concentration, right? And the solute concentration, if you are going to add more solute, what will happen? OP will increase, isn't it? And if you are not adding any solute, no solute is there, mind well, no solute means to say you are talking about the pure water, and for pure water, yes, my dear, OP is 0, mind well, OP is 0, isn't it? So this is a very important question may come to you, where OP is 0. Yes, this is for pure water having no solute, and by adding solute it will increase. We can understand them like that, okay? And yes, we can also measure this osmotic pressure okay, through the instrument, and that instrument is called as the, yes, osmometer. In our NCRT, diagram is given, information is given regarding potato osmometer. What is this? Very simple. We have taken the potato now. In potato, what happened? Just take it like this, okay, and make a pit. This is a pit inside, and see, this is a potato pit. Inside we have taken what we have taken. See, inside we have taken the solute, and due to that we have a solution outside. But outside, suppose only pure water has been taken. So what will happen? Outside, this is supposed to be the hypotonic solution. Inside, supposed to be the hypertonic solution. So water will move from hypo to hyper. Very simple. So suppose if they have got a particular layer, that layer will increase up. See, this is given here. See, this is your initial level, this is your initial level, and when it will go inside, what is going inside, okay, so what happened? Their level will increase, their increase up to here. So this is your final solution. Just osmosis, might just opposite, might be possible you are making this one as the hypertonic and outside hypo, means to say in the pit just put the pure water, and outside what happened? Just take the, yes, solution, just opposite. So what will happen? The second case will happen here. This is the second case. So what happened? Water will go outside. So there will be what? There will be the plasmolysis. So this is your initial level, and now this is your final level. Why it is decreasing? Because water is going outside. This is osmotic withdrawal, exosmosis, and this is your osmotic entry. This is the endoosmosis, right? Yeah, we can also understand very simply this potato osmometer through this experiment. See, I have taken here what? Three, we have the potato. Out of these three, one is boiled and two is not boiled, and one I am taking as a control. We are not using any difference in their solution. Both solution will be normal. One will be the hypotonic and other will be the hypotonic. So what will happen? Let's see. So we'll find here, see, this is a potato, creating the cavity here, pit it is, and see what will happen. Yeah, pit is there. So in the same way we will put three potato, but mind well, one is boiled one. So see, left side first one, what happened? It is boiled one. In middle, not boiled. Third one, right side, not boiled. And we are putting the first one as a control, first from right, and the left from, that is boiled, putting the salt in both the condition. Now outside we are creating the hypotonic solution by just adding pure water. So what will happen? Let's see. Let's see what is filled. Now outside we have the hypotonic solution, and inside what we have? The hypertonic solution. So see what is coming in the second case. See, in the second case what happened? Water has come. In first case, right side no water is there, means to say because they are supposed to be the isotonic solution it is right now, and the second case no, outside hyper, inside outside hypo, no solute, so hypo, pure water, and that's why it is moving from the low, that is the hypo, to hypertonic solution. And since it is membrane is being dissolved, being destroyed, so they are not working. So there is no role of the osmosis in the degenerated cell membrane, right? Yeah. So this is very interesting. Now let's see the third type, my dear, and this third type of this osmosis is called reverse osmosis, and it is used for the purification of water. What happened here? See, just I told you that in the first case I told you that we have created inside a very good what? Sucrose solution, and due to that water was not going inside. After that again increasing, water went inside. Now what I am doing, when their water is going inside, this is called endoosmosis, and osmosis was occurring that time. Now here what happened? External force is increasing, and due to increasing the external force, now what happened? Now they are not going outside, rather just balance. Again this is being increased. When it is going when again increase, so what happened? Opposite action is going on, means to say water is just moving from, yes my dear, from hypertonic solution to this pure water. Pure water supposed to be the hypotonic solution. So see how this osmosis is occurring, just reverse. This is what? Hyper to hypo. Earlier what we found? It was hypo to hyper. And why it is? It is due to the external pressure what we are exerting on the inner cellular system, isn't it? Yeah, inner solution. See this diagram, this is the fresh water and this is the salty water. But in this salty water supposed to be the solution. See, as I told you, this is the sucrose solution. So what happens when you are applying the pressure from the solution side, that is a salt concentration? So what will happen? This total bill supposed to be now, okay, it is hyper, mind well, and outside only pure water we have. So what we have, this is your hypo, and when they are increasing, see how only the pure water is going, and this is, you can say, this is the very good purification of water, isn't it? We do the same in the sea beaches, okay, because we are getting sea water, salty water there, and we are making their water purify through the RO. Okay, RO, you all know, in your house you are using, your every house nowadays we are using RO to get clear water, clean water, right? Yeah. Now again I want to make you more clear through the concept, my dear. So let's see the concept here. Yeah, see, again we have taken right side, we have the pure water, left side we have taken the solution. So see how they are moving from hypo to hyper. But when we create a pressure on the solution side, now let's see how we are going to create a solution on the pressure side, right side. So what will happen? Yeah, see, this is the pressure created. Now see how again water is moving from the hypertonic solution to hypotonic solution. So see, it's very much clear that we are going to purify the water, okay, from the dirty water to the very fresh water. So this is the role of RO, reverse osmosis, for purification of air, right? Now, so see, osmotic pressure, when we talk, this plant we have the leaf, here the maximum osmotic pressure. Why? You all know in plant we have the leaf, and what the leaf will do? Yes, my dear, leaf will perform photosynthesis, and due to the photosynthesis there will be glucose formation, and if there will be glucose, so what will happen? It will make them hypertonic solution. That's why they have maximum OP. In root we have the minimum, right? Now, yeah, now we talk about the osmotic pressure in different plants. Yeah, if you're talking about the hypotonic, they have 1 to 3 atmospheric pressure, xerophytes, okay, hydrophytes 1 to 3, xerophytes 10 to 30, mesophytes, that is your land plant, this is your xerophytic desert condition, sand plant it is, okay, this is your simple land plant, mesic condition we have, and this is your water plant, hydrophytes. So here it is 5 to 3, 15, and this is, see how this is 3 times more, right? 3 times more, and here see this is the 10 times more, very easy to remember. Now this is your halophytes, maximum, mind well, this is maximum. Question will come, where the OP is maximum? OP is maximum in the halophytes, saline condition. And when you create a hierarchy from the minimum to maximum, then you will move. Minimum we have the hydrophytes, yes, and maximum we have in the halophytes, hydro to halo, right? Yeah. So that's all, my dear, about this osmosis. We discussed what is osmosis, types of osmosis, osmotic pressure, reverse osmosis, and all, right? Now, so just enjoy just the same. Next class we'll discuss about turbidity plasmolysis, right? Now till now, yes, be update yourself with the Hortus Botanica. Prepare your this very important chapter, transport in plant. Every year 3 questions are coming in your NEET examination, isn't it? Yes. Now still we are running, the Omicron has come, so we have to be updated ourselves. We are now well experienced, we have 2-year experience of the corona, so we have to maintain ourselves and perform the best. Okay, so take care with your family members and all. Have a nice day.